All right, so again, I always think it is kind of useful to see, kind of take a look at this, and this is Instagrade. There's no like short answer thing. So whatever score you got on is the score you got. You know, this is always useful to see, like if you're down here, like at 50% or less, like realizing, oh, like that's not typical. Like you really need to be keeping up because we're going to have to be accelerating in this class because I kind of did finally kind of sat back and looked at what we got to cover and how much time we got. So, you know, there's still a lot to cover. So you don't want to fall behind. All right, sympathetic motor activity will. So basically the thing, you remember it's the fight or flight versus rest and digest. That's a pretty good mnemonic to kind of remember what sympathetic versus parasympathetic are doing. So the heart is going to increase. Sweat glands always increase. I talked about sweat actually being purely innervated by the sympathetic. Digestive processes are going to be inhibited, suppressed. So, you know, in general, again, it's this antagonistic thing. Like, you know, think about when you're kind of nervous or scared, your mouth gets all dry. You know, that's kind of suppression of, kind of digestive secretion. And predict, yeah, obviously dilate. That's something you probably should know for the, for our lab practical today. That was a big part of our, the pupillary reflex. Um, so somatic neurons. So if we are in the somatic motor division, it's going to be one neuron that goes all the way from the CNS to the target. We showed that. In the autonomic, however, we saw that there was a daisy chain that you needed at least two, but not at least, you had two. Went from the CNS to the ganglion and then from the ganglion to the target. So that's that. This one, blocking adrenergic receptors on the heart. So this one, you have to do a couple of steps of thinking. You have to think about, okay, adrenergic receptors. Adrenergic receptors, that is sympathetic, which means speed up the heart. If I'm blocking the adrenergic, I'm going to block the heart from speeding up. So that's what the answer here was, prevent the heart from speeding up. It most definitely will not speed up the heart because you're blocking the messages that speed up the heart. And it doesn't directly slow down the heart rate, right? Because it's adrenergic, the sympathetic isn't about slowing the heart down. What happens is it blocks the heart from speeding up, in which case the heart rate stays lower. But it's important to kind of understand the difference between directly slowing it down, which would be like a parasympathetic thing, versus keeping it from speeding up from sympathetic innervation. So, and again, these are really common drugs that are prescribed for high blood pressure, the beta blockers. They're also prescribed just for stage fright, so you can go in front of a crowd and give a talk and not spaz out because your heart's beating out of your chest and you're like, not able to focus. So neuromuscular junction, where somatic motor neurons innervate skeletal muscles, are characterized by the neurotransmitter, most people got it, acetylcholine, and the receptors, the nicotinic cholinergic receptors, the ones that bind acetylcholine and are directly gated, are always excitatory. And then here, remembering that I had fun writing this one. Remembering the word to answer this particular question is an example. This is kind of a self-reflexive kind of question. Declarative memory. That's like the stuff you can declare. You can say, you know what you know. Procedural is the stuff that you're learning, but you're not actually, exactly sure what you know, or but you have learned something that's, that you're using to kind of influence your choices. The other ones I just, I made up, rhetorical. Let's see. So any questions about those? Okie dokie. All right, so now we have one last part of the nervous system to go through, and that's the sensory. And yeah, we could easily spend the next 2 weeks just going over this, but I'm going to have to kind of speed things up a bit. So, I'm realizing there's just so much kind of interesting stuff that we can go into, and it's just on a practical level. We have to make sure we keep rolling along. So what we're going to do today is the afferent. So afferent, or sensory division. And like I mentioned before, there are both internal and external. What we call like the visceroreceptors, or interoreceptors. So there is a lot of the sensory stuff that's happening inside your body, like, stretch of your blood vessels, or the pH of your blood, or the your temperature, or all sorts of things, things that you aren't necessarily consciously aware of, but are important pieces of information that your nervous system uses to maintain homeostasis and, kind of keep your body running in order. We will talk about these, in turn as we go along. We have to talk about them when we get into the digestive processes or the blood pressure control. But we won't spend a lot of time right now talking about them explicitly. I will talk, the one I go a little more detail is like proprioception. The sensations you get that tell your brain the position and status of your physical body, your musculoskeletal system. The other ones are the external, the ones that you think about as like the five senses. This is where we're going to focus on for the most part. We're going to start with some general concepts around senses and receptors. And then we'll spend the rest of the time going into detail about the, more the eye, the ear, things that we've introduced in lab, but there's a lot more we should be looking at. But let's start with some of the just fundamentals first. So at the core of this all I mentioned in lab, and you should definitely understand this concept. Think ahead. Sensory transduction. So what do I, keyboard. So what do I mean when I say sensory transduction?
Converting the stimulus to an electrical signal.
Yeah, so you convert the good God, Mr. Autocorrect. Convert stimulus into electrical signal, for the nervous system. Right, so this is going to be a recurring theme. We're going to be talking about how do you convert light into electrical signals? How do you convert vibration into electrical signals? How do you convert the presence of sugar on your tongue into an electrical signal? That's going to be kind of the recurring theme. Is you've got some stimulus, and the stimulus is kind of the physical thing in the world. So when we're talking about, oops. Let's make it black. And this is like the physical energy. This is a real thing out there in the world. And then we also have the idea of your perception, like, your subjective experience. Subjective meaning like it's your thing that you're experiencing, what you believe is going on. And again, if you don't have a receptor that does the conversion of this physical energy into some electrical signal, you don't even have a chance of having a perception, right? The only reason we see is because we have cells in our retina that can convert light into electrical signals which feed into our brain, which go up through the thalamus into the visual cortex. Like I mentioned, the example I've used before, like, radio waves are things that are in the air that you don't see, you don't perceive because you don't have any kind of receptor that detects them. But they're there, for sure, because you take out a radio and you turn it on and you hear your music. It's like, it's got the instrumentality to convert that into something that you can notice. So when we think about this, I'll give you one more example, just because I think it's worth thinking about this. So in vision, you're looking at things. Good God, hold on a sec. I gotta go into airplane mode. Yeah, I apologize here. Okay. So when you're looking around and you see the wall, I'm looking in the rooms across here. I see, there's shadows. Some places, I know the room is white, but sometimes the white is bright white where the sun's hitting it, or sometimes it's grayish where it's more in a shadow. That, but you don't look at your wall in the sun and shadow. Or I look at Marissa's sweater and I don't think that she's got a sweater with these cool white stripes across it. I just think, oh, there's sunlight on there, but it's all one color that's more or less illuminated. But the reality, what's landing on my retina, the actual stimulus, the actual physical energy that is hitting my retina is very different colors, very different shades. But what my brain is extracting is not what is the color that's true, the veridical color. It's like, what is the probable pigment of the thing that's being illuminated? I don't really care, is it, what I really, so I'll show it to you. It'll be easier. This thing is one of the crazier examples of how your perception can be really different from what is actually like true. So here is this classic thing. There's like this little checkerboard with a cylinder. And if I ask you which square is darker or lighter, A or B, or are they the same?
A is darker.
Mm-hmm.
A looks darker. It's really, like, for me, it's impossible for it not to look darker. The reality is A and B are exactly the same color, except because of the way this is set up, where you kind of imagine the B is in the shadow, therefore it's going to be a little darker than it would be normally, and A is kind of in the sunlight, so it's going to be a little lighter than it would be. So your brain is making these calculations of, well, what's the true pigment, based on the lighting I'm perceiving, rather than what is the actual color landing on your retina. So I can show you. There's, if you actually connect A and B together, you can see here, they are identical. So the only difference between these two pictures is that little connector of gray between A and B. And even when they're both on the same screen there, and you're looking at them at the same time, like at least for me, I can't force my brain to see what's true versus what is kind of my perception of the pigment of the squares. So just kind of giving you this sense of, perception and stimulus aren't necessarily, your brain does a lot of kind of processing to figure out what's useful rather than what's necessarily absolutely true in terms of things here. So just to give you a sense, can anybody there actually force A and B to look the same when you look up there? It's freaky. I have to say, this one's kind of freaky to me. And again, because we don't have enough time, maybe I'll try to make a little page of little things for you to explore later on. There's so many cool things like this that actually let you see kind of how your visual system is working, and a lot of the weirdnesses of your visual system that you just aren't even realizing are happening, because you just kind of take for granted that you're seeing what you see, when in fact you are kind of making up a usable model based on the visual input coming in. So let's come back to the whiteboard. So this idea of perception, we have different modalities of stimuli that we have receptors for. So again, the receptors are the things that receptors do the sensory transduction. And the different forms of physical energy that we can convert into electrical signals and have a chance of perceiving, those are the modalities, the different forms of stimuli. What are the different modalities? What are some of the different kinds of receptors that we have?
Chemoreceptors.
So one are the chemoreceptors. So chemoreceptors are things that, what's the stimulus for a chemoreceptor?
Molecule.
And different kinds of molecules. So this would include your taste, your taste buds have chemoreceptors. So if there's sugar in your mouth. Your internal receptors are a lot of chemoreceptors. So what kind of food is in your intestines. So what kind of enzymes need to get delivered to break it down, or what is the pH, or what is the current sugar levels in your bloodstream. Do you need to go up or down there? So chemoreceptors, you have a lot of chemoreceptors, both internally as well as in the special senses, which we saw the smell and the taste are chemoreceptors. What are other kind of receptors that we have?
Mechanoreceptors.
There's mechanoreceptors. Mechanoreceptors, again, inside and outside. Inside, the mechanoreceptors are telling you that your stomach is stretched out and I'm feeling full, or are in the walls of your blood vessels, saying that your blood pressure is going up or down as the vessels stretch out. Or we're going to see inside your, joint capsules and stuff to say the angles of your joints at the moment. So a lot of mechanoreceptors inside your body, and then outside, this would be like your touch receptors, just stretch of the skin. Or like I've mentioned, hearing is actually a vibration of your eardrum. It's kind of like a touch right inside your ear canal there. It's like tick, 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 somebody tapping on your eardrum. That's what the air waves are doing. The pressure waves in the air are basically tapping, knock, 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 knock on your eardrum, vibrating it back and forth. And that's going to be detected by mechanoreceptors, the little hair cells that convert that motion into electrical signals. So mechanoreceptors, what other kind of receptors do we got?
Nociceptors.
Nociceptors. These were the pain. Holy crap, novice. That's crazy. I just got it. It wanted to change it to novice tots next. It's so weird. What do they detect?
Pain.
Pain.
Well, pain is the perception. That's what you feel. You feel pain, but what is the actual stimulus that is actually activating these nociceptors?
Tissue damage.
Yeah, it can be tissue damage or impending tissue damage. We're not going to talk about all of the signaling pathways, but when cells are being injured, they're going to release things that activate these nociceptors. Part of the inflammatory process is releasing prostaglandins that activate these nociceptors. So these are activated when either they're also, any kind of real intense overstimulation of any kind of receptor will start to be received as pain. But nociceptors, pain, there's something in danger of hurting, damaging, or in the process of damaging you. What are other kinds of receptors that we got?
Thermoreceptors.
Say it again.
Proprioceptors.
So I will write that here because people usually put it in here. Proprioception, though, I think of it as a sub category of mechanoreception, because it's basically mechanoreceptors that give you the position of your body. But I'll put it here as its own thing because it is a really important set of senses. We're going to talk about that in more detail in just a few moments. More
Photoreceptors.
photoreceptors. And yet there's even more.
Thermoreceptors.
Thermoreceptors. So all of these will take some version of a stimulus and convert it into electrical signal, whether it's molecules or physical deformation and stretch or vibration or tissue damage, or in this case, we're going to see is kind of a special case of this. Photoreceptors, light, thermoreceptors, temperature changes. The type of receptor that does it can be very different. So we're going to start with like this. Actually, let me mention, since we're going to, so get it out of the way, let me talk about proprioception. It's going to be the only of the kind of internal things that I'm going to speak about in more detail right now. Again, we're going to see more internal receptors as we go through the semester. But for right now, I want to explicitly introduce the proprioceptors. And then what we're going to do is go into the special senses. So let's do proprioception now. So this is basically giving you information about the current status of your physical body. There are three main kinds of receptors that are part of this. You know, you've met one already when we did the reflex lab. What is the stimulus for the patellar tendon reflex?
Stretch,
Stretch of,
muscle spindles,
yeah, exactly. So that we have the muscle spindles. Let me make it smaller. No. They detect stretch of the muscles. So, you know, I'm not going to get into the details of these. The muscle spindles are actually really cool little things that kind of reset their kind of baseline length, so like if the muscle has been stretched, they'll kind of readjust, so then they can detect stretch from the new position and all that. There's the intrafusal and extrafusal muscle fibers and blah, blah, blah. For this class, just know that muscle spindles are these cool little receptors in the muscle that can detect the stretch of the muscle. There are Golgi tendon organs. They're basically kind of telling you the tension, like how much tension, how strongly are the tendons getting pulled as they're trying to, the tendons are the things that connect between muscles and bones. So the muscles are trying to move the skeleton and the skeleton is resisting. And this is giving you a sense of the force that's being applied there. Joint angle receptors. Things that tell you the angle of your joints. They're actually just little stretch receptors. That's all they are. Like if you remember from anatomy, you've got a joint and most joints are synovial joints, right? What is around the outside of all synovial joints?
The capsule?
The joint capsule. Exactly. You've got this connective tissue capsule that is around the joint. And then the joint angle receptors, they're just little stretch receptors in there. Right, so as this joint moves one way or another way, if it's going this way, this will be less stretched, this will be more stretched. If the joint is, extending there, this one will become less stretched and this will become more stretched. So, it's not this magic thing to detect the angle of a joint. You just have to look at the relative amount of stretch of the capsule on either side of this thing to give you a readout on what is this joint doing right now. So these are really important. These become a major part of the input that the cerebellum is using to close the loop. You've got a sense in your premotor areas, the prefrontal, in the frontal areas, the motor association or premotor areas are having a motor plan of what you want your body to do. But then you need this information to actually carry it out, because you need to actually get feedback on what is my body doing and has it actually achieved the goal that I've set out to do and all that, right? So you need both. You need to know what you want your body to do, but then you need the feedback of what is my body doing and where is this current status and have I successfully done it? When proprioception is off, you get all sorts of weird stuff. You actually get weird things where people, when there's brain damage that affects this, it can really mess up just feeling like this isn't my body, like somebody's sewing this arm on me, but it's not my arm and stuff, because you're not getting the feedback of what it feels like to be your arm. People get convinced, like when I was asleep, someone must have, amputated my arm and then meticulously sewed on somebody else's arm that seems to work right, but it's not my arm. So you get really weird stuff. So it's important to, for also feeling embodied, that you have a sense of what your body's doing. All right, let's continue here. What's going on? All right, we're gonna now, let me think of, there's other big, all right. General senses versus special senses. So now we're going into the external stuff exclusively. We're going to be looking at, again, when you're kind of a kid, the five senses. And when you're a kid, you probably don't think about, oh yeah, lots of internal senses too, to detect the pH of my blood or something. But these should feel way more familiar. General senses we've been talking about. These are the things all over the body. These are the things that include like touch, pressure, temperature, pain. These are going to have very simple receptors. I'm going to be able to cover the general senses in just a cup. Wow, Morgan, that looks really cool with the sun behind you. You look like you're in some crazy movie or something. All right, so simple receptors, we're going to discuss them, but we'll go through them pretty quick. Special senses, here the receptors are in the head and more complex. This is going to be, vision, hearing, and equilibrium. Gustation and olfaction. Gustation, olfaction, hearing, equilibrium, vision. Each of these, like, luckily we've spent some time on these already, so I don't have to kind of start from scratch on them. But we could easily spend a lecture just talking about vision, or a few lectures talking about vision or hearing and equilibrium. There's lots of them. So we'll introduce them. If they're interesting to you, I recommend, you go deeper into them, because obviously I spent, that's what I did, went to grad school, was just sensory neurobiology. It's really fascinating trying to understand how you take in the world and make sense of it. So let's start with the general senses, because they're easy, or simpler, and then we'll shift into the special senses. All right, so like I mentioned, they're all over the body, and most of the receptors are literally just dendritic endings. Like here's the nervous system. You've got some neuron. The neurons tend to be kind of have their cell bodies. If you remember from anatomy, the dorsal root ganglion, the cell bodies of these neurons tend to be right near the spinal cord, the dorsal root ganglia. But then they just go all the way. And a lot of times the receptors are literally naked dendritic endings. So it's like the little dendrites at the end of this neuron have particular like receptors on them that are going to respond to the appropriate stimulus. A lot of like nociceptors are like this. Thermoreceptors are like this. You know, some mechanoreceptors, not all. So that's what I'm saying. They're simple. It's like if you looked at one of these under the microscope, there's no way you could tell what it is actually a receptor for. You would have to try different kinds of stimuli and see where it wakes up and starts depolarizing and sending an electrical signal. That's the only way you're going to know what the heck this thing is, right? It's not like you look at an eyeball, it's like, I know that's the thing that converts light into electricity. This thing just looks like little dendrites, you know, and like, let's make it hot. Nope. Let's like stretch it. Nope. Let's make it cold. Bzz. Ah, that's a cold receptor. So sometimes you've got some that have little encapsulations around them, little connective tissue around them. Some are encapsulated. You know, a lot of those touch receptors, like the little touch corpuscles, the Meissner's corpuscles, like Pacinian corpuscles, you know, they have some additional connective tissue around the dendritic endings, but they're still really simple. And we talked in, you know, so this is all, I think this is all I'm going to really say for the general senses. You know, and I'll reiterate the concept that we talked about in our lab, the idea of adaptation. What does adaptation mean?
Decrease perception with a prolonged stimulus.
Yeah, if there's a kind of a constant stimulus, they stop responding usually. So like we saw with the touch receptors, touch after a little while, just you can't even feel it anymore. Temperature just becomes kind of some generic temperature, you know, until you change it. And then all of a sudden you detect the change. Like I said, most senses adapt. Some of them adapt in more complicated ways. Some adapt literally at the receptor. There are there's a few senses that don't adapt. We talked about like pain receptors in general don't adapt, which, you know, makes sense because you don't want to ignore tissue damage, although it makes managing chronic pain really difficult because they don't ever shut up. Some of the things for like detecting your equilibrium and your kind of body position, obviously, you don't want that to adapt. You don't want to like sit still for a few moments and all of a sudden your body starts like disappearing from your self-perception. And it's like, whoa, I got to move again or else I feel like I become a disembodied spirit, right? That would be disconcerting to say the least. So like proprioceptors aren't going to do this adaptation, but most other senses do. It's also this idea, yeah, so there's other words, but let's just leave it there. Habituation is when you stop responding to something as strongly if you keep getting hit by it. So general senses. Yeah, it sounds like it's the person in charge of all the senses, but no, it's just more like generic, the things all over your body. Um, so the chemoreceptors for the special senses are going to be gustation and olfaction. Obviously, the molecules are going to be the stimulus, but where the molecules are is different. So, so gustation is looking at, molecules dissolved in your saliva. As you're chewing something up in your mouth. One of the parts of the lab you didn't do is like dry off your tongue and put sugar on your tongue. If your tongue is completely dry and you put a big pile of sugar on it, you can sit there for a minute, 2 minutes, 3 minutes, and you won't taste anything. In order for the taste buds to actually work, the molecules have to be in solution, have to get dissolved in the saliva, so then they can go and they can bind the receptors and all that. Olfaction, these are molecules in the air. Whenever you sniff, you're bringing air up into the upper parts of your nasal cavity where the little receptors are. And so it's the same basic idea. One's detecting molecules in your mouth dissolved in your saliva. One's detecting the little molecules that are, floating in the air. Hopefully not like commingling with the little coronavirus particles. In this case, we have taste buds that actually do the transduction. Here we're going to have this little thing, these little olfactory epithelium, kind of on the roof of the nasal cavity. In both cases, they have cells that bind ligands and convert it to an electrical signal. In your taste buds, there's going to be these little taste cells or gustatory cells. And they're going to have receptors. In the olfactory epithelium, you're going to have the little olfactory cells. They're going to have little receptors. So the basic idea is the same. The differences are where the stimulus is found, either in the saliva or in the air. And the other big difference is the kind of range of potential molecules that will actually cause a reaction. So let's, if we're looking at gustation, taste buds, they're basically like on your tongue, like maybe 80% on your tongue. There's also some inside your cheeks though, and down your throat, even like on your epiglottis at the very bottom of your throat. And the taste buds, I'm not gonna, if you've been in anatomy, you know more about taste buds. Taste buds basically have these little cells that are gonna, the cells with the receptors that can bind the ligand. They're going to have supporting cells. Both taste buds and the olfactory epithelium also have these little stem cells, the basal cells. They're called basal cells.
9 a.m.
And what do stem cells do?
Regenerate?
They
Make new
regenerate.
cells?
So I have these, and again, the taste buds are actually on the sides of the papilla and stuff. So I'm not getting into the details of the anatomy, but taste buds are in a very vulnerable position where you're eating stuff and things are scraping against them and you're eating hot stuff and this and that. I'm sure you have like destroyed parts of your mouth and tongue and the roof of your mouth, like eating different foods or Captain Crunch or whatever. So the life expectancy of one of these things is pretty short. So you constantly have to be replacing them and rebuilding them. So these little cells, these basal cells, there would be what we call like pluripotent stem cells. Pluripotent means there's a limited subset that they can become. They can become the different cells you need to rebuild a taste bud, but they can't make you a new liver or something. Right? These are not the stem cells that people talk about in stem cell research and all that. Those are like the totipotent stem cells. Those are the stem cells at the beginning of the development of the fetus that can have the potential to become any part of the body. So those are way more interesting in terms of trying to make a new spinal cord or something, or make a new organ. These are much more humble. They just rebuild the taste bud. And we talked about the basic tastes. The tastes tend to be the things that are useful to know about your chewing or what you're chewing up in your mouth. We talked about sweet being for like carbohydrates, right? Carbohydrates, we've, from the beginning of the semester, we see are kind of the essential way to get energy. Like if you need energy to survive, and the easiest way to get it is sugar. So it's really nice if you're chewing something up and it's like, oh, wow, I'm detecting simple carbohydrates. That's awesome. This is going to be a great source of energy for me and keep the engine rolling along. Salt, electrolytes, we've seen that you can't have any of the electrical signaling in your nervous system if you don't have adequate amounts of potassium and sodium and chlorine and all of that. So that's essential. Remember the word salary comes from salt. So it used to be so precious. People would actually get paid in salt because it's essential for being healthy and makes your food more pleasant too. If you don't have enough of those electrolytes, like some of the people, like there's been a few of those cases where people get extreme with this. They read something like, I need to drink like 16 glasses of water a day to be healthy. Maybe I'll even be healthier if I drink 30 glasses of water a day or whatever. And people get into these conditions. There's like hyponatremia, where you don't, or your sodium is way too low, or hypokalemia, where your potassium is way too low. And then all of a sudden your nervous system starts working right and your heart stops beating right and you die of a heart attack or something. So you need to have adequate electrolytes. So that's another good thing to be able to detect. Oh, there's some salt in there. There's sodium and chloride. That's essential for the functioning of a healthy nervous system. So I want to eat that. Bitter. I've mentioned alkaloids. A lot of plants have molecules that they use often to protect themselves. They often do interact with our nervous system in ways, or our body in ways that we want to pay attention to. A lot of the alkaloids are poisonous. Some of them are, poisonous depending on the dosage. Some of them we use, like coffee is an alkaloid, or caffeine, I should say, is an alkaloid. But in general, if you have something and it tastes bitter, that's telling you that there is some alkaloid type molecule in there, and you might pay attention because it might have some effect on your physiology. Sour is just low pH. People now talk about umami as another flavor. This is like kind of glutamate. When people talk about monosodium glutamate, flavor enhancer, accent, it's activating umami detectors on your taste buds. It's kind of like this beefy flavor. It's nice. This is another thing. If we were in lab, I always have a little shaker of accent, which you can just buy in the grocery aisle. It's just pure MSG. MSG is actually, for almost everybody, is not a bad thing. It's no worse for you than just eating. You don't want to eat too much. It could be like eating too much salt or something, but it's good. I sprinkle it on my tongue and I'm like, yum, that's really nice. And most people are actually fine with it. They've done some tests where they checked people who were convinced that they are deathly allergic to MSG. When they actually look at it for real, it has no effect on them at all. Not everybody. There are people that genuinely can have a reaction, but so I'm not going to say, but I can say in general, it's got a bad rap. So umami, like when you're fermenting stuff, the gluten, these things like, soy sauce and stuff has a lot of this kind of stuff. So gustation. And like I mentioned, this is just part of what we think of as taste. Because when you have a cold and you can't breathe, your taste buds are working just fine. And still the food, quote unquote, tastes really bland. I can barely taste my food. It's like you're tasting it, the gustation is working just great. What's missing is what? What's missing in that deeper sense of appreciating your food?
Olfaction.
The olfaction. Whoops. So in your nasal cavity, up on the roof are these little olfactory cells. They actually send their little axons up through. Does anybody from anatomy remember what are the little holes in the ethmoid bone that the olfactory nerves go up through?
Cribriform plate.
The cribriform plate. And then they go into this part of the brain called the olfactory bulb. Which then is going to just continue on into the rest of the brain. Again, these have receptors on them. Like I mentioned in lab, so I won't do again, there are all sorts of ligands, molecules that are recognized by these receptors. Again, some of them you have just like if there is sodium chloride on your tongue, you have the perception of salty. Or if there is, sucrose on your tongue, you have the perception of sweet, right? So you have the experience of sweet, but what does it really mean? It means there are sucrose molecules dissolved in your saliva. The same thing, different molecules that come in here. It's like, oh, it's lilac, it's butterscotch, it's dog poop, it's like sulfur, it's hyacinth. Those, what you think of as the experience of a smell is just the presence of a particular kind of molecule that is being detected by your olfactory epithelium, these olfactory cells that are sitting here and that have all these receptors that detect different shaped molecules. And people have really spent a lot of time looking at olfaction it doesn't seem like there are core smells that you then build other smells from. Like, taste, there's kind of these basic tastes. For smell, it seems much more of this big continuum. People have spent a lot of time trying to understand kind of the more subtleties of the sense of smell, and it's complicated. Things about smell. Olfaction. This is the only sense, that doesn't have to go through the thalamus. But every other sense we've been talking about, it comes into the thalamus and then gets distributed up into the cortex. Olfaction seems to be kind of the most ancient of all of the senses, and it has projections that go into the hypothalamus, into the amygdala. It's connected in these much more primitive ways into your emotion, into your memory circuits and all that. So it's fundamentally different than the other senses, which are kind of more abstracted in some ways. They go up through the thalamus and up into the cerebrum and processed in this kind of higher level way. This can get down into the deeper brain regions directly. Smell. I mean, it makes sense that it is kind of the most ancient. If you think about what it is telling you what kind of molecules are floating around in my environment, right? Whenever you sniff or smell, you're basically sampling what is this milieu. I am moving around here through space, and what is around me. And again, if you were some primordial bacterium, some little single-celled prokaryote living in a primordial soup, 3 billion years ago, you still need to have the sense of smell. You still need to know, like, is there something that I can metabolize for energy? Let's move and tumble towards it so I get in the midst of it and or is there something that is noxious and is dangerous for me? Let's tumble away from it so I stay safe, right? So it's like some very core functioning of sampling what is your environment, what molecules are in your environment, and how should I respond to them. So the other thing I will mention, because it's hard not to, the idea of the vomeronasal organ. So there is like, so in these detect pheromones. I'm going to just do this really briefly because there's not much time, but I do want to just introduce this idea. So the story for a long time had been in non-human animals, including a lot of non-human mammals, in addition to the olfactory sense, up in the nasal cavity, at the top of the nasal cavity, there are also receptors that are not about detecting molecules to be perceived as smell, but instead detect molecules that are then actually, they're more like like hormones that go from one individual to another. So you have, one individual here giving off some molecules, and then another individual over here, and they're like sniffing, and it goes up, and their nervous system can detect these little molecules that are being given off by this organism, by this individual. So it's kind of like inter-individual signaling through molecules. And in a lot of non-human animals, it's very clear it's related to mating behaviors and things like that. So the story was like lots of other animals have them, but humans are much more sophisticated, aren't being influenced by molecules being given off by the other person. And more recently, both anatomically and physiologically, there's evidence that we do have something of this system. Um, and that we do actually detect things giving off by other individual humans, and that it can affect us in ways that aren't necessarily as obvious, like, oh, I'm smelling something. Instead, it can affect behaviors or perceptions. Like one of the studies that was interesting was looking at, they had like people smell shirts, and which shirts, people had worn a shirt for a certain amount of time, and then they were in little boxes, and then you'd smell the different shirts and which one did you like, which one didn't. And what they found was actually this correlation between kind of genetic relationship. And we're going to talk about MHC later on when we do the immune system, but it's like kind of your cell ID marker that makes your cells you, MHC. They found like the shirts that were the most like pleasant, and it wasn't like the ones that were like sexy, oh wow, this makes me horny or something. It was more just like, no, this, I like this makes me comfortable. I like this. And this one, like, nah, yeah. And this one, yeah, no, that's nice. The ones that people that made that it was a positive association tended to be ones where the genetic separation was more like, it was actually a mix. So there was like kind of MHC similarity versus like, positive, positive experience. And basically when the MHC was too close, it was not a very positive experience. As there was more genetic dissimilarity, it became more positive. And then they found as it got really different, it kind of went down again. But what it seems to be, at least, and this is speculative, but it makes sense to me, and it is kind of compelling, is that we are broadcasting things to each other that are giving information about kind of compatibility. You don't want to mate with somebody who is really, really genetically similar. That's when you start getting into all sorts of genetic defects and stuff. That's why you're not supposed to like have kids with your sibling, right? Is if it's much more likely that you're going to get two bad copies of a bad gene. So you are doing this without even meaning to. If you're just hanging out with somebody and they're really close in terms of MHC, you're more likely not to like have that same experience of like, oh yeah, this is it. I'm really feeling it, versus, so if it makes more sense on a kind of genetic level to mate, you're more likely to have this sense of like, it's more positive association. And obviously this is one thing out of a million other things, but the fact that it is there influencing or coloring your experience is fascinating to me. So just kind of putting that out there, that there is evidence that, again, it's still, there's evidence that we do something like this, and it's not just non-humans. They're kind of, a lot of the research are kind of going back and forth. There's been all this stuff about, humans versus the animals, which is so stupid because obviously humans are animals. We're just, we're the mammals. So more and more we're realizing humans have a lot of things that we've said are just, the non-human animals, and they're finding more and more a lot of non-human animals have a lot of the things that we liked to make us super special in terms of emotions or cognitive functioning and problem solving and all that. So that's been kind of nice. I think there was for a long time, I think it's just kind of this don't know, this weird thing where, God created man as a super special thing and all. It's like, we were just part of this evolutionary process and became another mammal. And it's pretty clear when you look at the evidence that, there's just kind of this continuum of mammals with different properties and different behaviors and abilities and all that. So that is the chemoreceptors. The other ones now that we have to do, and actually so we are not, I am aware that exam 2 is going to have to be pushed ahead a week. So actually, I'll just say that right now, because there's no way exam 2 is going to be on this coming Tuesday, because I'm not going to finish everything I need to finish in the next 15 minutes. So just, putting that out there. And hopefully that does not ruin anybody's plans.
So instead, it will be on the 3rd on election day.
Yeah, so that way, if you are, depending on how it goes, either you're going to be blind drunk celebrating or blind drunk trying to like not like shoot yourself. So, but either way, it's that you'll have it done before you have to like look at what happened. So so that is a little, I mean, kid grief. We'll do it. I think it'll work out. I mean, the fact is, if you're not distracted, you're probably just anxious and distracted now anyway. So it's not like going to be that much different in a week from now. All right. So now we're going to do vision. So vision. So we've been spending time looking at the eyeball. In fact, hopefully you know some things about the eyeball because it's going to be on your lab exam. Obviously, we talked about the eye. You could look at a cross section of the eyeball. You can look at the eyeball from the front. Let's make this person with green eyes, the green iris here. And the iris kind of is this little, it's made out of smooth muscle, kind of create the pupil, which is a little hole that the light comes in. Then we also talked about, you have a little lens behind that so you can adjust the focus. The retina, where you actually, what happens in the retina?
Sensory transduction.
That's where the actual sensory transduction is happening. That's where you got the rods and the cones that actually convert the light into electrical signals and send it out the optic nerve. So this part we've looked at, and we've actually looked at it more in terms of visual acuity with the emmetropic eye and hyperopic eyes and myopic eyes and things like that. Other things that I should bring in for kind of completeness for the eyeball, let's talk a little bit about the flow of aqueous humor. So just like we saw the CSF in the brain, where you're constantly producing CSF and draining CSF, and hopefully that is working well, or else your brain's going to start inflating. I just made my head disappear. How did I do that? You have this constant production. So actually, I can back up just a second. The cornea, which you've met, is clear. The lens, which you've met, is clear, but obviously they have cells. And obviously you don't want to have blood vessels going across the cornea or across the lens, because then you are going to, it's going to obscure your vision. So you have this fluid that is being produced and is being drained that kind of irrigates everything and keeps everything kind of getting what it needs. So I didn't mention the word for these before, but now I will. These little structures that both anchor the lens and have the muscles that adjust the focus, as well as are going to be creating this aqueous humor. These are called the ciliary bodies. So they have the little muscles, the little ciliary muscles that adjust the lens, but they also create aqueous humor. So it's this fluid, I'll make it like blue, that kind of fills the front of the eyeball. The back part of the eyeball is actually filled with this jelly stuff called vitreous humor. Maybe I'll make it, I don't know, blackish. So this stuff I just drew here. Vitreous means glass-like. It's kind of like a gooey gumdrop. It kind of is solid in there. So your eyeball kind of holds its shape and all that. It's being kind of irrigated by the aqueous humor can kind of percolate in there, but it's not like just a swimming pool back there. It's more just like a gooey jelly back there. So the aqueous humor, though, is flowing freely in the front of the eyeball. And it's being produced constantly by the ciliary bodies, which means we need to be draining it, or else the eyeball is going to start inflating and build up more and more pressure. So the drainage for this aqueous humor is going to be through these little canals, the little canals of Schlemm. Act, that is ugly. In this picture, just to make it more clear, it's right at the boundary between the cornea and the iris. You're going to think of it as like this thin donut. Right? We're looking at a cross section here. That's why it looks like just two little circles above and below, but it's all along that boundary between the iris and the cornea. And so you have this constant production of aqueous humor from the ciliary bodies and a constant drainage into the canals of Schlemm, which then ultimately drains into that whole like kind of jugular vein as well, right? It's just more kind of plasma-like stuff that gets dumped back into your general circulation. what is the medical condition if that flow is blocked and you start building up intraocular pressure?
Glaucoma.
Yeah, that's glaucoma, which can be really dangerous. If you have that pressure for extended periods of time, it actually starts damaging your retina in the back, and it can permanently damage your vision. Like when they take you in for an eye exam, you've probably noticed that they don't just check your visual acuity. They also actually check your intraocular pressure. They have little things to make sure that that pressure is right and that you don't have that buildup in there, because that, like I said, that can cause long-term damage to your vision. So what do we call it if the lens gets clouded?
Cataracts?
That's cataracts. It is possible if people have cataracts, they pop in a new lens. What's going to be the problem of surgically just putting in an artificial lens in somebody's eye?
That it cannot be arranged by
Yeah,
the muscles.
exactly. You lose the ability to accommodate. So now you're basically either always going to be in distance vision or always going to be in close vision, and you don't have the ability to adjust the shape of the lens. So sometimes people get both eyes set for distance vision and then just have to use glasses when they look at something close. A lot of people actually get one lens, they get the two different lenses in the two different eyes. So they have one eye that has got fixed focus for distance and one eye that has fixed focus for close. And eventually the brain just starts using the correct eye and taking the image in depending what they're looking at. And to me, that doesn't sound like how I'd want to go, but I do know people who have that and say they at some point they don't even realize that they're doing it anymore. So, but yeah, so that's the problem with, it can change your life. Like my mom had cataracts and was terrified of going under surgery and everything. And like then that afternoon, like I was checking her out because I was checking how she was doing and thinking that it was going to be traumatic. And she was like, just so thrilled. It's like, oh my God, that's amazing. I can see everything so clearly. It's like, it was crazy. Because eventually people start just looking like everything's through this really kind of just gauze. And it's, so cataracts. Corneas are also things that are actually easy to transplant because corneas have no blood vessels. When you have your little organ donor card, and they go harvest you for stuff, they will most definitely take the corneas. I had one guy in my anatomy class that was one of his jobs was harvesting organs from freshly deceased people. And this was the first thing they go for because you can transplant them very easily. So anyway, and then all right, it's 9:30. Let me think if there's anything else I want to say about the eyeball, because the rest of what we're going to talk about is in the retina, the part that's actually doing the sensory transduction. But because the eye is kind of a more complicated organ, I wanted to get into some of this other stuff as well. The iris, we could just briefly mention again. Iris under autonomic control. There are muscle fibers that are circular that are going to constrict the pupil. What division of the autonomic nervous system is going to activate the circular fibers to constrict the pupil?
Parasympathetic.
Yeah, parasympathetic, right? So if you want to give somebody a drug to do an eye exam so you can shine a light in their eye and the pupil doesn't shut down, what kind of drug would you give them? And it's eye drops. They don't have to take it systemically. You just put
And
eye drops.
acetylcholine antagonist.
Exactly. Cholinergic antagonist. That's gonna block that pupillary constriction in response to light. There's also longitudinal muscles that are gonna be sympathetically activated to dilate the pupil, pull it more wide open. And the pupil does affect focus and different things. And they change with convergence. So there's more to it than we're going into, but basically they are important for adjusting the amount of light coming in. They do affect depth of field and stuff like that as well, but we're not going to get into that. What else? Floaters are just, everybody's seen floaters. Those are just like little bits of stuff that are in there that you see. Depending if they're closer to where the retina is, they're going to be more in focus. Sometimes you even see them as kind of stuff. They're further out, they're just more like blurry, kind of there's something there. All right. It's 9:36. All right, so I want to make sure we have enough time to do the exam and all that, and also check in for a few minutes before the exam starts. So let's take a break. Let me double check my Canvas here. As of this morning, when I was about to start the class, there were a number of people who still hadn't turned in their table of contents. And make sure everybody's done that now. One. There are 2 people who have not done that. That has to be turned in. So please do that. And we'll meet back at 10 o'clock. At 10 o'clock, I'll do any last questions. We'll talk about just some of the details, and then we'll just start the lab exam.